if your body's sodium-potassium pumps just stopped working right now.
Oh, you'd be in serious trouble. Like, immediately.
Right. Every single cell in your body would literally swell up and burst within minutes. I mean, you wouldn't bleed out, you wouldn't suffocate, you would just dissolve from the inside out simply because this microscopic protein engine forgot how to move salt.
Yeah, it's wild to think about. Because, when we study human anatomy, we usually just get this beautiful static snapshot of structure. But anatomy is just the structure. Physiology, on the other hand, is the function. It's the movie of human life playing out in real time.
Exactly. It relies on these incredibly delicate balances of, pressure and electricity and chemistry just to keep you alive. And today, we are pressing play on that movie. Welcome to our deep dive. We're unpacking some fascinating source material today from the USMLE Roadmap Physiology.
Which is such a great resource. And our mission here is really to act as your educational guides. We're putting together a comprehensive, high-yield masterclass on human physiology.
Yeah, we're going to connect the dots, going from the microscopic, like the lipid bilayer of a single cell, all the way up to these massive systemic cardiovascular and renal functions.
Yeah.
And we're tailoring this specifically for you.
Right, whether you're a medical student prepping for boards or maybe a young professional in the health sciences or, just an intensely curious learner who really wants to understand this biological machine you're piloting every day.
I love that.
Okay.
So, let's unpack this. Because before we can look at the whole systemic machine, we have to sort of isolate the individual building blocks. And the sources start us right at the border of the cell, which is the plasma membrane.
Yeah, the classic fluid mosaic model, which basically means it's not some rigid brick wall. It's a dynamic, really flexible layer. And the foundational structure is the lipid bilayer.
Right, which relies entirely on the amphipathic nature of these lipids. They automatically organize themselves into this barrier. Right.
Exactly. The membrane is the ultimate gatekeeper. And the specific composition of those lipids actually matters immensely. I mean, phospholipids are the most abundant. They kind of form the bulk of the sea, so to speak.
But then you also have cholesterol in there, right?
You do, yeah. Cholesterol inserts itself right between those phospholipids. And its job is to regulate the membrane's fluidity and really improve its structural stability. And then you also have sphingolipids, which are heavily concentrated in nervous tissue.
Actually, I was looking at the clinical correlations here regarding those sphingolipids, and it really highlights how devastating just a tiny microscopic lipid imbalance can be.
Oh, absolutely. Because when those sphingolipids are modified, like by adding carbohydrates, they become glycosphingolipids, which are super vital for brain cell function. But if a person had a genetic defect...
Like where the lysosomes can't produce the right enzyme.
Exactly. If they can't produce the enzyme to break those glycosphingolipids down, they accumulate. And they become totally toxic inside the neurons. This is actually the underlying biochemical mechanism of Tay-Sachs disease.
Wow. Yeah. It leads to progressive paralysis and really severe impairment of mental function. It just shows how a failure to manage a single type of membrane lipid can cascade into a catastrophic systemic collapse.
It really puts the stakes of cellular structure into perspective. I mean, it's not just abstract biology. Now, nestled within that fluid sea of lipids are all these proteins. And when I visualize this fluid mosaic model, it feels kind of like a heavily guarded, super exclusive nightclub.
I like that analogy.
Right. Like the lipid bilayer itself is this massive bouncer. It keeps the general uninvited crowd out, mainly water-soluble molecules. But the integral and peripheral proteins, they act as the VIP doors.
That's a perfect way to look at it. The integral membrane proteins physically span the entire lipid bilayer. They often use these alpha helices to anchor their hydrophobic segments right in the core of the membrane.
And the ratio of bouncer to VIP door changes depending on the cell, doesn't it? It changes drastically. Take myelin, for example, which insulates nerves to speed up action potentials. It's less than 20% protein. It needs to be mostly lipid to act as a really effective electrical insulator.
Oh, that makes sense.
But then if you look at metabolically demanding cells, like in the liver, their membranes were more than 60% protein just to handle the massive constant influx and efflux of metabolites.
Right, they need more VIP doors. But getting things through those doors often requires energy.
Which brings us to primary active transport. This is where the cell uses ATP to just force substances across the membrane against their natural concentration gradient.
Which brings us right back to that pump you mentioned at the very beginning of the deep dive.
Yes, the sodium-potassium pump.
Yeah, that single transmembrane, ATPase, basically dictates the survival of the cell.
Hmm.
It uses ATP to pump 3 sodium ions out of the cell and 2 potassium ions into the cell. And by doing that, it accomplishes 2 huge things.
Okay, what's the first one? Well, first, because it moves a net positive charge outward, it maintains the resting membrane potential. But second, and honestly more importantly for immediate survival, it regulates intracellular osmolarity.
Because water follows salt.
Always.
Always. If sodium builds up inside the cell, water is going to rush in to dilute it and the cell bursts. So the continuous extrusion of sodium offsets that natural osmotic pull.
It's just bailing water out of a sinking boat constantly. And alongside sodium, the cell goes to these enormous lengths to manage intracellular calcium, right?
Oh yeah, it sequesters it incredibly tightly within the sarcoplasmic reticulum. The cytoplasm is kept virtually devoid of calcium, just totally empty of it, until a very specific signal arrives.
And when that calcium is finally released into the cytoplasm, it acts as this massive biological trigger. Like in the myocardium, the heart muscle, that flood of calcium is the direct signal that initiates muscle contraction.
Exactly, it flips the switch.
Yeah.
Which is actually a brilliant transition, because once the myocardium contracts, we're moving from the cellular building blocks to the macro level, the systemic pump and the pipes.
Right, the cardiovascular system. And hemodynamics is what dictates how that pump and pipe system actually operates. The physical relationship governing this is pretty straightforward. Velocity equals flow, divided by cross-sectional area.
Meaning, if the cardiac output, the flow, stays constant, the speed of the blood is inversely proportional to how wide the vessel is.
You've got it. So blood moves incredibly fast in the aorta because it's just one relatively narrow pipe for all that volume. But as it branches out into millions of microscopic
The
capillaries,
total combined area gets huge.
massive. The total cross-sectional area of the capillary bed is enormous. And the physiological brilliance of that design is that the blood velocity drops to an absolute crawl once it hits the capillary.
Which is exactly what you want, right?
Yeah.
It gives it time.
Exactly. That deceleration provides the necessary time for diffusion. Oxygen, carbon dioxide, nutrients, they all need time to trade places between the blood and the interstitial fluid.
It's like a rushing river fanning out into this really wide, slow-moving delta. Now, driving all that flow is the pressure gradient generated by the heart. And the sources point out something really interesting about pulse pressure, which is the difference between your systolic peak and your diastolic resting pressure.
Right, there's a very distinct mechanical change that happens there as we age. A widening pulse pressure is a classic hallmark of aging, and it's due to decreased vessel compliance.
Meaning the pipes get stiff.
Exactly. The arterial walls become stiffer, they lose that elastic recoil. So when the left ventricle ejects blood into a really stiff aorta, the systolic pressure just spikes aggressively. The vessel can't stretch to accommodate the volume.
Oh, wow. And then what happens during diastole when the heart is resting?
Well, because there's no elastic recoil to maintain the pressure, it just plummets rapidly. So the gap between your systolic and diastolic numbers gets much wider.
That makes perfect sense mechanically.
Yeah.
Now, to understand how the heart actually modulates its output to deal with these pressures, we have to look at Starling's law of the heart.
Ah, yes, a fundamental principle.
Right. The idea is that increased preload, which is the volume of blood stretching the ventricular wall right before it pumps, results in an increased stroke volume. So the incoming blood physically stretches the muscle fibers, and they just snap back with greater force.
The underlying mechanism there relies on the physical alignment of actin and myosin filaments inside the sarcomere. When you stretch the muscle fiber, you're optimizing the overlap between those filaments so they can grab onto each other better. Exactly. More cross bridges can form, which generates a much more forceful contraction. It's an automatic system that ensures the heart pumps out whatever volume of blood it receives from the veins.
But let me push back on this a little bit, just to map out the physical limits. Because if stretching the myocardium makes it pump harder, does it just scale infinitely? Like if venous return massively spikes, will the heart just keep stretching and pumping with the force of a cannon?
That's a great question, but no. Starling's law has a strict physiological limit. A normal heart operates exclusively on what we call the ascending portion of the Frank-Starling curve. Up to a certain optimal stretch point, yes, more volume equals more force.
But what happens if you go past that point?
If the sarcomere is stretched beyond that optimal length, the actin and myosin filaments are physically pulled too far apart. They actually lose their overlap.
Oh, so they can't grip each other anymore.
Right. Fewer cross bridges can form, and the force of contraction just sharply declines. Clinically, we see this in conditions like severe dilated cardiomyopathy or decompensated heart failure. The ventricle is chronically overstretched, and its pumping efficiency just plummets.
Man, that's fascinating. To see these volume mechanics in action during daily life, let's talk about positional changes, something everyone experiences. What is actually happening hemodynamically when you suddenly stand up from laying down on the couch?
Well, the second you stand up, gravity causes roughly 500 to 700 milliliters of blood to just instantly pool in the highly compliant veins of your legs.
That's a lot of blood just dropping to your feet.
It is. And this immediate venous pooling causes a sharp drop in venous return to the right side of the heart. So following Starling's law, decreased stretch means decreased stroke volume, which leads to a transient temporary drop in your mean arterial pressure.
Which is that split-second sensation of feeling lightheaded when you stand up too fast.
Exactly. But your body catches it almost instantly. Arterial baroreceptors, which are located in the carotid sinus and the aortic arch, they detect this drop in pressure within milliseconds.
And they sound the alarm.
They do. They decrease their firing rate to the brain's cardiovascular centers. The brain responds by withdrawing parasympathetic tone, taking its foot off the brake and unleashing a massive surge of sympathetic outflow.
The gas pedal.
Right. This instantly increases your heart rate, increases the heart's contractility, and triggers peripheral vasoconstriction. It restores your arterial pressure before your brain is dangerously deprived of oxygen.
It really is an autonomic symphony.
Mm-hmm.
And while those macro pressures are adjusting, there is this microfluid balance happening right at the tissue level, guided by Starling's capillary forces.
Yes, the capillary tug of war.
Exactly. It governs fluid exchange across the capillary walls. You've got hydrostatic pressure, which is the physical force pushing fluid out of the capillary. And then you have oncotic pressure, which is the osmotic pull generated by plasma proteins, primarily albumin, pulling fluid back into the capillary.
And any disruption to either of those forces has immediate clinical manifestations, like prolonged standing. That elevates the hydrostatic pressure in your lower extremities simply because you have a column of blood fighting gravity.
So the pressure just physically pushing the water out of the pipes.
Yes, it forces excess fluid out into the interstitial space. And if that exceeds the capacity of your lymphatic system to drain it, you get dependent edema, swollen ankles.
Right. But conversely, we see edema driven by the exact opposite mechanism in patients who have suffered severe burns or who are facing starvation, because they lose those vital plasma proteins.
Exactly. Without enough albumin in the blood, capillary oncotic pressure drops severely. The blood essentially loses its osmotic grip on the fluid, so the fluid just leaks out into the tissues.
Okay, so we've pushed all this blood into the capillaries to deliver oxygen, but the body has this incredibly elegant mechanism for knowing exactly where to drop that oxygen off.
Oh, this is one of my favorite concepts. It relies on the oxyhemoglobin dissociation curve and a phenomenon known as the Bohr effect.
Yeah, the Bohr effect basically dictates that the curve shifts to the right in specific environments, which means hemoglobin's affinity for oxygen goes down. It lets go of it easier.
Right. And those specific environments are characterized by high carbon dioxide, increased acidity, meaning more hydrogen ions, and elevated temperature.
So the local chemical environment physically changes the shape of the hemoglobin molecule.
It does. It alters its allosteric conformation, which stabilizes its deoxygenated state.
Which is just brilliant when you think about it. I mean, imagine hemoglobin as a delivery truck. If it's driving through a quiet, resting tissue bed, it holds on to its cargo of oxygen pretty tightly.
Because the tissue doesn't need it urgently.
Exactly. But when that same truck drives into a skeletal muscle during an intense workout, an environment that is generating massive amounts of heat, pumping out CO2 and building up lactic acid, the truck naturally just throws its cargo out the window much more aggressively.
Yes. It's an automatic, highly localized offloading system that reacts perfectly to the metabolic demands of the tissue it's passing through.
It's amazing. But, managing the waste generated by all those metabolic demands requires a totally different system, the kidneys.
The kidneys are incredible. They regulate the extracellular fluid, which makes up roughly 35% of our total body water, compared to the 65% held inside the cells.
And the sources talk about assessing renal function clinically using the concept of clearance, which, as they point out, isn't a measurement of how much stuff ends up in your urine.
No, it's not. Clearance is actually defined as the volume of arterial plasma that is completely cleared of a substance per minute. We use the clearance of very specific markers to accurately estimate the glomerular filtration rate, or GFR, which is really the ultimate benchmark of kidney health.
Right. But the kidneys aren't just passive biological Brita filters, right? They are an active control center for systemic blood volume and pressure. And they do this primarily through the renin-angiotensin-aldosterone system, the RAAS cascade.
The RAAS cascade is crucial.
Let's trace how a drop in blood pressure actually triggers this massive hormonal response.
Okay, it begins at the macula densa. This is a specialized cluster of cells in the kidney's distal tubule that physically sort of taste the sodium chloride concentration of the fluid passing by.
Like little microscopic salt sensors.
Basically, yeah.
Yeah.
If systemic blood pressure drops, the filtration rate in the kidney slows down. Slower flow means there's more time for sodium chloride to be reabsorbed earlier in the nephron. So by the time the fluid finally reaches the macula densa, the salt concentration is exceptionally low.
And that triggers the alarm.
Right. That low salt signal triggers adjacent cells, called juxtaglomerular cells, to secrete the enzyme renin directly into the bloodstream.
And renin is what sets off the chain reaction. It cleaves angiotensinogen, which is a protein that the liver is just constantly pumping out, into angiotensin I.
Yes, and then as that blood flows through the tiny capillaries in your lungs, an enzyme called ACE, or angiotensin-converting enzyme, converts it into the active hormone, angiotensin II.
And angiotensin II is powerful. It acts like a biological tourniquet, right?
Oh, very powerful. It causes intense peripheral vasoconstriction to rapidly elevate your blood pressure. But simultaneously, it also stimulates the adrenal cortex to secrete another hormone, aldosterone.
And what does aldosterone do?
Aldosterone acts on the collecting ducts in the kidney. It upregulates sodium channels to massively increase sodium reabsorption. And because water obligatorily follows sodium, that water gets pulled right back into the blood. This expands your total fluid volume and secures a long-term recovery of your blood pressure.
But retaining all that water requires the kidney to heavily concentrate the urine. And the mechanism driving this is countercurrent multiplication in the lift of Henlo, which, reading through the sources, just feels like a masterclass in biological engineering.
It really is. The architecture of the loop is the key here. The descending limb is highly permeable to water, but not to salt.
Okay.
But then the ascending limb is the exact opposite. It uses active transport to aggressively pump sodium, potassium, and chloride out into the surrounding tissue, the medullary interstitium, but it is completely impermeable to water.
So it's trapping all this salt deep in the kidney tissue, creating this massive osmotic gradient.
Yes. The kidney spends energy to make its own tissue incredibly salty.
That's wild. So then when antidiuretic hormone comes along and opens water channels later in the collecting ducts, that ultra-salty tissue acts like a dry sponge. It just passively sucks the water right out of the urine and back into the bloodstream.
Exactly. The active creation of a gradient drives the passive conservation of water. It's brilliant.
It really is. We have all these incredible systems, transmembrane pumps, baroreceptor reflexes, the RAAS cascade, oxygen offloading. Coordinating all of this chaos requires the ultimate command centers, the endocrine and autonomic nervous systems.
Right. The endocrine system uses chemical messengers to maintain homeostasis. Water-soluble hormones, like peptides, they can't cross that lipid bilayer we talked about.
Because they're repelled by the hydrophobic core.
Exactly. So they have to bind to extracellular membrane receptors, which then activate intracellular second messengers, like cyclic AMP, to actually execute the physiological response inside the cell.
And cells actually possess a really vital defense mechanism against overstimulation here, known as downregulation, don't they?
They do. If a target cell is constantly bombarded by super high concentrations of a hormone, it will actually internalize and degrade its own membrane receptors.
It physically removes the VIP doors.
Right. It physically reduces its own sensitivity to the signal just to protect itself.
Looking at specific endocrine cascades, the whole process of insulin release from the pancreatic beta cells perfectly illustrates how multiple cellular mechanisms link together. It's like a cellular Rube Goldberg machine.
It really is. So glucose enters the beta cell via a transporter called GLUT2. Once inside, that glucose undergoes metabolism, generating ATP.
And that rise in intracellular ATP is what closes the ATP-sensitive potassium channels on the cell membrane, right?
You've got it, which prevents potassium from leaking out. That causes the cell membrane to depolarize. This sudden change in voltage opens voltage-gated calcium channels.
There's that calcium trigger again.
Always the calcium. That influx of intracellular calcium causes the vesicles containing insulin to fuse with the membrane and dump their contents into the bloodstream.
Glucose metabolizes to ATP, which closes potassium channels, alters voltage, opens calcium channels, and triggers insulin release. Boom. Now, on the other end of the metabolic spectrum, the sources heavily detail thyroid hormones. They act synergistically with the sympathetic nervous system to increase your basal metabolic rate.
Yes, but there is a crucial developmental window here that the sources emphasize regarding congenital hypothyroidism. Thyroid hormone is absolutely essential for normal brain development and myelination in infancy.
Right, the stakes are so high there.
Unbelievably high.
Right.
If a newborn lacks thyroid hormone, they will develop irreversible, severe intellectual disability, which was historically termed cretism. The critical window for intervention is just within the first month of life.
Wow, just 1 month.
Yeah. If thyroid hormone replacement is started before that window closes, neural development actually proceeds entirely normally, which is exactly why mandatory newborn screening panels are implemented worldwide.
That structural necessity brings us nicely to the hardwired control center, the autonomic nervous system.
The hypothalamus serves as the master integrator, right? Particularly for temperature regulation.
Right. It triggers either heat conservation mechanisms or heat loss mechanisms, primarily sweating, depending on what your core temperature is doing. And the autonomic nervous system is divided into 2 branches, the parasympathetic rest and digest branch and the sympathetic fight or flight branch.
Which brings up something I found fascinating. A seeming contradiction here when looking at thermal stress. Sweat gland activity is explicitly controlled by the sympathetic nervous system.
Correct.
But if the sympathetic system is triggering a fight or flight survival response, like preparing you to physically fight a bear or run away, why is it initiating sweating? I mean, shouldn't a survival mechanism conserve water and energy rather than expending it just to cool down? It's a great question, but you have to look at what the sympathetic nervous system is actually doing. It isn't just a panic switch. It orchestrates total body optimization for intense physical exertion.
Okay, so it's prepping the engine for a red line event.
Exactly. If you engage in life or death combat, your skeletal muscles are going to generate massive amounts of thermal energy. If your core temperature exceeds critical thresholds, your cellular proteins will actually begin to denature and you will collapse.
Oh, so it's proactive.
Very proactive. Sympathetic stimulation preemptively triggers sweating to ensure you survive the thermal stress of the exertion itself. And furthermore, this pathway is a really unique physiological exception.
How so?
Well, almost all postganglionic sympathetic nerves use norepinephrine as their neurotransmitter. But the sympathetic nerves that innervate your sweat glands are cholinergic, meaning they release acetylcholine, which is normally the neurotransmitter exclusive to the parasympathetic system.
That is so cool. It's incredible how every microscopic anomaly really serves a precise systemic purpose. We've journeyed all the way from the amphipathic lipids forming the cell's gatekeeper to the active transport pumps dictating cellular volume.
We've watched actin and myosin stretch under Starling's law to maintain cardiac output.
We've seen hemoglobin dynamically alter its shape to offload oxygen exactly where it's needed and trace the RAAS cascade from a single salt sensor all the way to systemic vascular clamping.
It's a lot to take in, so to solidify these principles, let's step into the role of the clinician with a quick review exercise for you based on today's masterclass.
Let's do it.
Imagine a patient arrives in the trauma bay having suffered severe, extensive burns. This has led to a massive loss of plasma proteins from their bloodstream. Based on Starling's capillary forces, which specific pressure hydrostatic or oncotic is primarily altered here? And will this alteration favor fluid moving into the tissues or fluid reabsorption back into the capillaries?
Okay, let's reason through the mechanics of that capillary tug-of-war. If you lose the plasma proteins, like albumin, you lose the osmotic pulling power. Therefore, the capillary oncotic pressure drops severely.
Right.
So without that inward pull and force, the hydrostatic pressure pushes outward unopposed, which favors massive fluid filtration out into the tissues, resulting in severe edema.
Spot on. Applying those foundational physical laws allows you to accurately predict really complex clinical realities.
Beautiful.
It is. And as we conclude, I want to leave you with a final thought regarding that cellular defense mechanism we discussed earlier, downregulation. We established that cells actively decrease their surface receptors when constantly bombarded by an excess signal just to protect their internal homeostasis.
Yeah, pulling away the VIP doors.
Right. Well, think about how this elegant microscopic defense mechanism might scale up to human behavior. You know, are the neurological pathways in our brains actively downregulating their dopamine receptors in response to the constant artificial sensory overload of the digital age?
Man, That is a provocative thought. It really makes you wonder if that numbness we feel after scrolling on our phones for hours is just our own physiology trying to protect us from our technology.
It very well could be. The body is always adapting, always seeking that balance.
Always. Well, keep questioning those balances, keep observing the machine, and we'll catch you on the next deep dive.